So, so good evening, everyone. Welcome to the first rendezvous session of this semester. So today we have Dr. Jyotishman Dasgupta with us. So before introducing him, I would like you all to know that I have been working with him since the beginning of the year, and I know him past 3.5 years when he first came to CBS to deliver his rendezvous session personally. So this introduction is going to be a bit personalized. So Dr. Jyotishman Dasgupta, as people call him in TIFR and everywhere, JD. So he's an associate professor at Department of Chemical Sciences, TIFR Mumbai. He has been associated with TIFR past 10 years, and he's mentoring students like a parent, guardian, and a friend. So I have been in the close proximity with him, and I have seen how he is mentoring his students, and uh, and the values he give. But he has gone through since his academic career, and he's a very enthusiastic person, and always tries others to engage in discussions and question as much as possible. He keeps the environment friendly, enjoyable, joyful, laughable, and scientific. So, without a further ado, I would like JD sir to start the session. Thank you.
Okay, well, this is.
Sorry to interrupt. Just before you start, I mean, people feel free to ask questions. Just raise your hand before you want to speak anything, and you can, like, I think it's fine with sir that you interrupt.
Also, it's fine if they directly interrupt me. It's okay. Okay. Cool. Guru Sahib, thanks for making me blush. Okay, and but I seriously want to thank you and all the.
So your mic got muted.
Okay, sorry. Yeah. So so I would like to thank Guru Sahib and all the rendezvous organizers, especially the CEBS Science Club, if that's correctly spelled out, for promoting this talk in social media. This is pretty big for me, it really is. It's always been exciting time when I have interacted with CEBS students. And it's really a deep honor for me to give this talk in this forum for the second time. And notice I used the word time. And incredibly today I am going to talk about a chemical reaction clock. Okay, and this is a pretty strange title. You would ask, what does that really mean? Chemical reaction clock, what does it mean? What is it about? Well, the answer is actually given in this slide in some sense. Okay, you can see a molecule. Can you see my cursor? Just say yes if you can.
Yes, yes.
Yeah. So you can clearly see a molecule here. I'm not going to name the molecule right now. It's just a molecule for you. And just as we look at human beings and when we look at molecules, we only care about the physical characteristics initially, right? Or the chemical constituents of the molecule, the atoms, right? And as with human beings, once we see their pictures or we come in close contact or proximity, we have a conversation, we get to know the person. And we almost, while making a conversation, we almost remember, sometimes we say, literally, this guy has a heart, right? And an associated heartbeat. So, so molecules too, beyond their physical description, have something called a vibrational beat. Okay, and all of you never imagine that when you first see the molecule, but deep within it is this motion, which is always happening in time. Okay, and the entire colloquium is going to be focused on how do we keep a tab on this heartbeat or a vibrational beat of the molecule as the molecule is going to do a reaction. Okay, so that's part of the colloquium, and that's part of what I'm going to sort of emphasize today, that watching these beats can help us do a novel classification of chemical reactions, which are beyond what the textbooks give. Okay, But let's first start at the very fundamental, at the very basic. When we say chemical reaction clock, it ends with the word clock. Let's go and see what a clock really means, right? So this is just a picture I took from Google. One of the wall clocks that you would see, and strangely, all wall clocks give the same time in a picture. And there are many stories behind it, but maybe one of you know better than me. We can have a discussion after the talk. But if you were to see a wall clock, you see the minute hand, the hour hand, and sometimes it's not very visible here, the second hand. And what does the clock do? It is a device that measures and keeps and indicates time, okay? And it has a fundamental unit for its operation. The clock always has a fundamental unit for its operation. It has to keep time. So time has a unit. In this case, we use the second hand as a unit to characterize time, right? And we hear the ticks of the clock all the time when we are close by the wall clock. The second hand moves and there's a ticking sound, right? So how does it all relate to reactions? So what is a chemical reaction clock? Well, let's first go into what is a chemical reaction. This is for everybody. You have studied through your high school, through your schooling in the chemistry textbooks. Well, the definition of a chemical reaction is nothing but it's a process that encompasses rearrangements of constituent atoms of the reacted molecules. Okay. In this case, I'm showing a very interesting reaction of coming due to a collision between carbon monoxide, which is a gaseous molecule, and dioxygen, another gas. When they collide, they can make CO2, a linear molecule, and a gas that we exhale. And it also, because of the rearrangement of atoms, it gives a nascent oxygen. This is all in the gas phase, of course, but that's an example of a very simple chemical reaction. But notice how I introduced the chemical reaction. I did not say that the chemical reaction is defined by only this particular scheme that is shown below. I have kept another scheme which does not lead to chemical reaction. And there is a reason for it, okay? In this particular collisional process, the reaction did not take place, which tells us from this particular image that not all degrees of freedom that are available to the molecule, that are translational, rotational, or vibrational, will lead to a chemical reaction. So it's a puzzle. The puzzle is set by the fact that when I look at any reaction, and reactions are important, I'll come to that, can I understand by which degree of freedom a molecule chooses to react? And does that lead to selectivity? Okay, so that is sort of the puzzle that we as chemists always sort of seek in our daily research or our daily thought process or our daily education, right? But typically when we go through the field of chemistry, the subject of chemistry, the way it is taught, we define chemical reactions under categorization, subcategorization of the kinds, the classes that are associated with either an electron movement, like a redox reaction. Like a redox means it has to be sort of broken up into reduction and oxidation. RED for reduction, OX for oxidation. So reduction and oxidation processes, they are clubbed together because they involve electron change, right? They involve removal of electrons or dumping of electrons into system. So that is the classification, one class of reactions. When you do not have electron change, you might have exchange of atoms. And the simplest form of that is an acid-base reaction, right? For all redox reactions, I've given an example on the left-hand side here. Photosynthesis that we all know of is an example of redox reaction, right? We have CO2 getting reduced to carbohydrates and water, H2O, getting oxidized to oxygen, right? So now, for acid-base reactions, things that we do in the lab, in the chemistry lab, I don't know how many of you did the practicals where, you were used the litmus test to know pH change. You could see colorimetric changes on your litmus paper. The litmus paper is actually undergoing an acid-base reaction. And when it's protonated, okay, it changes its color. When the litmus paper material, the chemical in the litmus paper is deprotonated, it again changes its color. So it's a wonderful chemical reaction that shows up in your eye. Okay, and we'll talk about how our eye perceives color, but pretty shortly. But this is essentially a chemical reaction that is visible to the naked eye. And of course, the last class of reaction where there is no atom exchange or electron removal or electron dumping or electron removal. The last class belongs to changes in the structure. So you already have a molecular system, and that molecular system reorganizes itself to one kind of structure, another kind of structure. Proteins do that all the time, right? They have an unfolded structure and a collapsed structure. So biologists in this audience will connect very well. The folded structure of a protein is much, much more significant than the unfolded structure most of the times. Well, of course, there are certain proteins with unfolded structure and they have a huge biological importance. One of the classes, of course, amyloids, right? There are these unfolded proteins which play a critical role in biological function as well. So rearrangement reactions are a class by themselves. They allow for us to talk about structural changes within given existing manifold structures. I think Komal has a question. Komal, do you want to ask a question? Okay, maybe she's having a problem with the audio.
I have a question.
Who is, okay, can I see you in the video?
This is Pratyush, yeah.
Pratyush, okay, hi.
Yeah. Okay, the host has stopped me from sharing my video.
That's okay.
Anyway,
I know how you look.
So my question is, so in this slide, like all the terms that you've written that are there are terms that I already have studied and can relate to, like redox reaction, acid-base reaction. But I didn't know that electron transfer reactions, like can we classify redox reactions as reactions that have electron transfer and acid-base as reactions that have proton transfer? Because is it that conclusive?
Oh no. So you're right. These are examples of those, sub-examples. Okay. When I say redox reactions, acid-base reactions, or rearrangement, those are the major classes. Okay, Pratyush. Yeah. And electron transfer reaction. A redox reaction need not just be an electron transfer reaction. It can be associated with an oxygen removal or a hydrogen removal, but it always has an oxidation state change, which is related to counting of electrons. Okay, Pratyush? Yeah. So, and similarly, acid-base reactions, the proton transfer reaction is one class of acid-base reaction. You can do other kinds of acid-base reactions as well without involving a proton. Okay, so these are just one sort of representative examples of reactions under the major class. Okay, Pratyush?
So are the redox reactions and acid-base reactions, these major two major classes, are they actually completely different from each other, or there is an overlap also?
No. There will be combination of these. These are classifications. These are traditional classifications. But real reactions will involve all three of them. Maybe I will show you an example. That's a good question. We'll discuss that.
Okay.
Yeah, like a hydrogen atom abstraction reaction, OH getting broken into O and H, right? That means that electron has been taken and a proton has been taken.
Yeah.
So that has to be classified as a redox cum acid-base reaction, if you can classify it step by step.
Yeah, that makes sense.
Yeah. So these and it's important we have this conversation because this sets up the pitch for the talk. Because You really need to understand that we are going to be absolutely fundamental in our discussion. Okay.
Okay.
Yeah. So now, all of these are reactions, and one of the things that we need to notice ourselves is that a combination of these reactions are what makes us humans, right? And it's essentially how chemical reactions at the very fundamental level are connected to each other in reaction networks in our body. That constitutes our biological sort of function, and that also tells us how we are thinking, how we are processing information, how we are delivering information. So all of this is really fundamental to our own existence. So understanding the reaction networks in our body, what a biologist does, and some of you are interested in, is really critical. And some of you say biological reactions, but ultimately they're all chemical reactions, okay? It's just associated with biology, yes, but at the fundamental, when I say molecular interpretation of biological mechanisms, it really means chemistry. You're dealing with chemistry. You're dealing with reactions. You're dealing with reaction networks, okay? This is one example. This is nothing but acidification of water by CO2. I have mentioned this because this is the primary source of something that we all experience when we don't have food for a long time, acidity, okay? The fact that CO2 can be solubilized in water and form something called carbonic acid, okay, at very low pH. This particular acid has a very sort of transient existence. It really, people have not really, they've done some spectroscopy on carbonic acid under low temperature, but it has a very, very short lifetime. It immediately dissociates into a proton and a much stable bicarbonate ion in our body. Our body pH is typically referred to be at around close to 7.4. And biologists can tell me more about it. There are pockets in our body where things are acidic, slightly more acidic than 7.4, but typically the bulk pH averages out to be close to 7 to 7.4. And bicarbonate is pretty stable under those pH conditions. And hence, it's a more stable system to exist in. Hence, the proton is lost and the equilibrium is pushed towards bicarbonate. And that, in a sense, is the governing equation in your body that regulates your acidity, okay? Your physiological buffer is carbonic acid and bicarbonate equilibrium, okay? And whatever you do, when you metabolize something, you're going to acidify your gut pH or whatever. You sort of, the buffering action of this is critical to maintain the pH. And if you don't, then of course you have all these symptoms. Some of them I get because I typically am prone to a lot of acidosis. It's kind of interesting that this reaction really does not happen naked in our body, okay? It really takes into account that there are some molecules that ensure that this reaction is controlled. In some of the organelles in our body, cells, there is something called an enzyme called carbonic anhydrase, okay? And this particular enzyme is there, its existence is there simply because CO2 plus H2O going to carbonic acid is not an isolated reaction in our body. It's a reaction that is connected, coupled to many other processes and metabolic pathways. And hence, a regulation of the time and the reproducibility of the reaction has to be maintained. And for that, nature has designed a wonderful enzyme called carbonic anhydrase. And what it does is it has a zinc metal. You can clearly see zinc 2+, a very interesting element in our periodic table. It really does not belong to the transition metal, but it still is. It's sort of an outcast. It's been used here for doing this reaction. Why? Well, CO2 is a gas molecule. Water is liquid, right? In all its being. So if I had to do a heterogeneous reaction of a gas molecule and the liquid, I can only I have to fight against the entropy of the gas. I have to keep the molecule in contact with the liquid all the time. For doing that, the enzyme actually plays an important role. It takes in and solubilizes both the gas molecule and the liquid water. And then all of this sort of phase part goes away, vanishes. When you are a single molecule, you really don't care about phases, right? You're just a single molecule sitting inside the protein active site. And then you're doing the reaction. It is reproducible. It's actually concentrating the CO2 in presence of H2O. And this reaction happens this way, that H2O first gets activated on the zinc. It loses its proton because the pKa of H2O, which is close to 15, actually, typically you would say that it's around 15.4. It gets reduced to pH of 10 or near abouts when it's bound to the divalent zinc, because it's +2 charge. It actually allows for the water to lose one of the protons and becomes hydroxide. Hence, the pKa of water gets affected, and it becomes a zinc 2+ hydroxide system. And then once CO2 comes in, right, sitting there, comes in and reacts with the hydroxide, it forms bicarbonate. It's nothing but CO2 plus hydroxide is bicarbonate. There is no redox. It's not a redox process. It's an acid-base reaction. And then you just lose the bicarbonate in the cycle of the enzyme by pushing in water. Okay, so you basically remove proton, you deliver bicarbonate, and then you recycle the enzyme. And that's what the entire acidification cycle with CO2 of water is done by carbonic anhydrase. And this is done in the reverse as well. The enzyme can work both ways. Okay, so why this is done is simply because there's a cycling going on, and you would have a timing circuit associated with the cycling. You want bicarbonate to be delivered in a repeated period so that your metabolic pathways are working in conjunction. So these are time-ordered reaction networks. And I think Sanskruti has some questions, maybe.
Yeah, there's a question on chat. So, does the enzyme via zinc facilitate diffusion of CO2 and water, or does it also lower the TS?
Oh, that's of course, that's a very good question. The enzyme does both. It's not just the job of the enzyme to concentrate things, but it does. But apart from that, the entire activation process of water reacting with CO2, it lowers simply because it deprotonates first, and the hydroxide actually reacts faster. If you were to do this reaction in your own home, you can do this. You make your water alkaline. Okay, you would see, and if you have, say, for example, silver salt or something, or barium heavy metal salt, you can make carbonates. You can just precipitate out carbonates after a few days. Okay, and you will see the precipitation rate of formation of carbonates in alkaline water is faster than in regular neutral water. So this is, of course, the transition state goes way down simply because you are now doing the reaction with hydroxide instead of doing it with water. You never form carbonic acid in this. Okay, so that's the idea. Okay, so now so let's take one thing at a time. I said that the timing of these reactions are critical, and enzymes are there to provide that timing, the clock for these reactions. Why? As somebody asked, the transition state gets lowered and you increase the rate of the reaction, correct? And that's what is shown here in this plot. But let me first introduce this plot to all of you. what this plot means. This plot is actually energy. Hi Savita, is there a question? I think Savita may not have a question. Okay, yeah. Okay, so so let's look at this plot. So the energy axis, it's essentially total free energy of your reaction, and there is a reaction coordinate axis which shows progress of the reaction. That means on the left side I have the initial state. On the right hand side, as I'm moving from left to right, I actually look at progress of reaction. So this is the final state. This is essentially a plot to show protonation of an anion X minus. Okay, it means that the initial state, the proton is far removed from X minus, and then after using its degree of freedom, the proton can translationally diffuse, or it could be held by an enzyme, or it could be held by somebody else, pre-organized. Then the proton is right there at the transition state to do the reaction. And then once the proton moves over and forms a bond, it all goes downhill and you form a very stable HX molecule. Okay. So this particular free energy profile gives you a very simple idea of a reaction, but in a thermodynamic perspective. Okay. What you get is energies of the original initial state, which you can actually look back in the y-axis, plotted back in the y-axis, and the final state. Again, you put it back, project it back on the y-axis. The gap between them is the free energy change of the reaction. Okay, is the delta G. The more negative it is, the more favorable the reaction, right? But also what you see is something of this point, which is the transition state. It's called the transition state, which defines the degree of freedom that the molecule used. How did it use it? In which orientation did the proton approach the X minus? And that is the qualification of being a transition state. It doesn't stay there for long, it just drops out and it forms a stable molecule. Now, this transition state also has a y-axis. It has an energy associated with it. But what is more important is the characteristic of this transition state is defined by the x-axis, which is the reaction coordinate or the progress of the reaction. And I will come to this a little bit later, but yes,
sir, I have a question.
Yes, Jay, please.
So you related degrees of freedom. So can we relate actually? So if a molecule has more degrees of freedom of interacting, say H has more degrees of freedom with interacting with X, for example. So will that reduce or increase the energy? I mean,
that's a great question.
Proportional.
So when I give you these kinds of plots, these are reaction energy profiles. Okay, these are called reaction energy profiles. What it means is I am monitoring a reaction and I'm plotting the energy profile of the reaction. That means the initial states of the reaction, the final states of the reaction, and the transition states. These are the three constituent parts of the reaction energy profile. Okay, so Jay, based on my x-axis, the question is loaded from your side, because what is my choice of the x-axis is important. Not all degrees of freedom lead to a reaction yet. Okay, as I said in the very first slide of a chemical reaction, not all degrees of freedom will lead to a chemical reaction. It's a puzzle that you have to solve. Only few such degrees of freedom will lead to the reaction. So this plot is only going to be connected by a smooth line if this is the reaction coordinate or the reactive degree of freedom. Okay, all other reactive degrees could be existing. All other degrees of freedom could be existing, but they are not reactive degrees. Okay, so let's assume for a simple system, n atoms, right? You have 3n degrees of freedom, right? We have all learned this from basic physics. An atom can have 3 degrees of freedom. A molecule with n atoms can have 3n degrees of freedom. Now, once it's a molecule, there are only 3 sort of degrees of freedom for the translation, right? And the 3 for rotation. And the rest is divided into how atoms are connected to each other by vibrations, right? So the thing is, out of those 3n, Jay, Any of them could be a reaction coordinate. It could be translational diffusion, that means translational motion. It could be rotational motion, or it could be a vibrational motion. So only in one of those or few of those you can draw these reaction energy profiles. And does that answer your question? It does not matter if you add on degrees of freedom, it may not change your reaction energy profile. But now, if you add on degrees of freedom that actually contributes to the reactive degree, then Jay, yes, I can sort of change these numbers of these three constituent states. Okay.
Sir, this is Prithvish.
Yes.
Sir, I wanted to ask that is it free energy versus reaction coordinate or enthalpy versus, because
and what enthalpy? Oh, yes, I can do it as enthalpy also, Prithvish. But if I can measure free energy of reaction, why don't I plot it as free energy versus reaction coordinate? It's okay.
Yeah, if it is a free energy versus reaction, I am ignoring the kinetics factor of this reaction. I am not looking into the transition state and all. But if it is the free energy profile, then let's say HX is an acid, right? It's a weak acid.
Right.
Okay, so we can write that HX reversibly to H plus and X minus in a normal aqueous medium.
Yes, yes, yes.
So in that case, doesn't the free energy delta G become 0 in case of that, when that reversibly?
No. The free energy of any reaction is defined by the free energy of the reactants, subtraction of the free energy of the products, and subtraction of free energy of the total free energy of the reactants. Okay.
Yeah.
So in reversibility, in the condition of reversibility, the free energy is 0. Okay. What it means is what you have done is at some point, okay, when you have established relative populations to be equal, you can keep them at equal energies. Okay, that is when equilibrium is established. However, it's not the plot here. This plot is the true free energy that you change from one as you go from the reactant to the product. Okay. Okay.
I got it.
Okay. So now, I don't know if Sanskriti had any more comments. Sanskriti?
Yeah. So, how do we know which degree of freedom would react? I mean, is it theoretical or do we find it using experiment? So
that's the question that I'm going to address. Yes, it's experimental, and there's a lot of computations associated with it as well. At this point, Sanskriti, it's a myth almost, because experimentally only few molecules have been looked at whose reaction coordinates or reactive degrees of freedom have been identified by chemists all around the world. Okay, but the reason I'm giving this colloquium is simply because the textbook knowledge of ours about classification of reactions needs to be reordered, simply because now there are more experiments and computations that sort of tell us that if you can find the reactive coordinates of a molecule, you have done enough about the reaction. You have known enough about the reaction. So you don't need to classify, because every reaction is different and you are identifying the key step of it. So the classification becomes moot because you can address all reactions individually. Okay. So it's almost like individual diagnostics, right? Medical society is going towards personalized healthcare, right? We don't want to classify people as these people have chronic neurological disorders. We classify, right, based on symptoms, right? But where we are moving at is personalized healthcare, which means every person is different, slightly different, and you would like to treat a person based on their characteristic. It's almost like that. Looking at chemical reactions, you have a classification because you never had the knowledge of reaction coordinates, but because now things are changing, maybe it's the idea of classification should be debunked and every reaction be treated separately, right? So that's the idea. Okay, so let's move forward and ask the question. So what does this free energy diagram give you? Typically, these give you, apart from the energies, a rate. Okay, a rate of the reaction. Why? Because the initial state and the transition state have an energy separation. And by Arrhenius model or Arrhenius theory, you can calculate rate of a reaction by just taking the exponential of the separation between these two state energies. Okay, and that becomes an important parameter for any reaction. So the question then becomes, looking at free energy profiles, can rates be a discriminator of reaction types? Because that's what sort of will be critical, because rate is important, right? I told you the enzyme maintains a rate. The enzyme maintains a network connectivity, right? So rate is important. However, can rate be a discriminator of reactions? No, simply because a proton transfer reaction or an electron transfer reaction can have same rates. Okay, so that is where I emphasize, you guys have put in the questions together. I emphasize the x-axis, the reaction coordinate, and I give you an example how it's important to look at the reaction coordinate. So this is an example. I think Pratyush, you were asking about combination of reactions, and I had right in place a mixture of two simple reactions in this example, which is called an H-atom abstraction. An H-atom abstraction reaction can be looked at as a proton transfer combined with an electron transfer. Why? H-atom is nothing but an electron plus a proton. H plus an electron. Everybody is with me, right? So H-atom reaction is a very simple form of a combination of redox event and an acid-base event. Okay, so let's look at how how typically people think about it. Okay, so this is an example, very simple reaction. A reactant RCH3 on the left. If you can see my pen now, RCH3 moves to RCH2 dot, right? So what I have done, I have removed a hydrogen atom from this. RCH3 is a hydrocarbon, typical hydrocarbon. It could be ethane, it could be methane, whatever. I have taken out an H atom and I made it an RCH2 dot, a product of a reaction. But see how I've constructed my free energy profile or energy profile, whichever Prithvish wants. Prithvish, you can choose anything. You can do enthalpy, you can do free energy. I don't have any problem associated with it. I have drawn it in 2 dimensions. Did you see this? First one is the electron transfer reaction coordinate. The second is a proton transfer reaction coordinate. So I have imagined H-atom abstraction in 2 steps, 2 sequential steps. One is a redox event, this one, and the other is an acid-base event. Okay, and both of them have their own reaction profiles. You have a minima for the RCH3, you have a transition state for the electron transfer event, you have a minima for the intermediate radical cation, and then again a transition state, and now you end up into the product, right? So this is the reaction coordinate for a sequential proton and electron transfer step. However, you can be clever, chemists can be clever. They can say, no, I don't want to think about it as separate events. I know H atom is moving as RCH3 is going to RCH2 dot. So somebody is picking that H atom in totality together. So what is that class of reaction? Well, you can imagine that if they are coupled, the electron and the proton transfer events are coupled. That means this particular coordinate becomes minimalistic single coordinate. Okay, you've opened up this second dimension and you projected it back to the single dimension and said, aha, you know, there is a dimension, that there is a coordinate that exists, part of the degree of freedom of the system, that allows for one kind of a reaction energy surface. Okay, I don't have to imagine 2 different coordinates, and that's a coupled reaction, that's a cascade reaction. Okay, and that goes to an intermediate. That intermediate is this radical cation, which is in between. But now you see the energy profiles are different. In this coordinate, you can actually gain rates And this really happens in systems, in biological systems, where biological systems use something called as proton coupled electron transfer reactions to catalyze reactions and make it faster. You could, in the original diagram, if you remember, these barriers could be larger, these individual barriers could be larger, but then in the new diagram, these barriers are much smaller, and you can sort of see the changes, see the drop in the energy. So that means the rates increase in certain places of coupling with the electron and proton together and doing this entire process in one go. Okay, so or in coupled manner. A hydrogen atom transfer actually is a very special case of a coupled proton and electron transfer process, but most of the times it could be just pure coupling of the electron and proton transfer events, which sometimes we do in the lab. I'll get to it at the end. So,
sir, I have a question.
Yes, John.
So when you do this from going to 2 dimensions to 1 dimension, is this like a something for mathematical ease, or can you measure all these intermediates? Which
that's fantastic question, John. Yes, in principle, this is for our own simplicity of the framework to understand the framework. But yes, this question of coming to a reduced dimensionality for the reaction coordinate is not simply a mathematical thing that you're doing, but you are actually invoking coupling between the electron coordinates and the proton coordinates in some sense, right? And there are experimental verifications for this. One of the verification for this exact thing is what you can do is you can separately do the electron transfer reaction. Okay, John, you can separately do the electron transfer reaction, calculate the rates here. And you can do the proton transfer separately, again calculate the rates here based on the transition states. But when you do it together, you do it much faster than that. And the other thing is that you can use a heavy atom effect, that means a deuterium effect. So you can replace the proton with a deuteron, and in the single go you will observe a kinetic isotope effect, which cannot be explained by a simple two-step reaction, but it has to be a single-step reaction. So that's an excellent question. Yes, people have observed the evidence for a reduced dimensionality using rate experiments as well as proton versus deuteron isotope measurements.
For experiments, how do you separate out the electron and proton transfer coordinates? Like they always happen at the same time, right?
No. So they don't. You don't separate out. In a reduced dimensionality, you don't. It's together. It's a new reaction coordinate, and it involves both charge transfer or proton movements as well as electron separation. And it's through the molecule. The molecule is going to show you the picture.
In the chemical reactions, they always happen together, right?
Means?
No, these are like describing some chemical reactions. So these two reaction coordinates happen together, right?
Yes, happen together. Absolutely. The product is formed in a concerted manner. Yes, John. Yeah.
So, but how can you separate them out as separately? How can you separately visualize electron transfer and proton transfer?
You can't. So what you do is, John, you ensure that when you're comparing it to the electron transfer and proton transfer coordinates, you have to stop the reaction of the radical cation. You really have to technically stop it. Okay. And then you do the radical cation reaction separately. So in the coupled sense, you set up the system in such a way that you have no control on that, right? It will happen at one go and reach the neutral radical form. But in the case when you separately would like to do it, you have both an electron acceptor and a proton acceptor present along with the system, separately with the system. So you, in one case, you have proton acceptor and electron acceptor and proton acceptor both for the concerted reaction. In these separate cases, you only have a proton acceptor and an electron acceptor separately for a reaction. Okay, John. So that's how you separate out the reaction rates. You do measurements of concerted processes and compare the free energies.
So, so there are a couple of questions.
Yeah, sure.
So, Vatsal wants to ask if all this is a mathematical artifact or is it something real?
maybe Vatsal needs to probably ask this question in the video, because this is a little bit of a difficult question for me to understand. What does he mean by mathematical artifact?
Actually, his mic is not that good, so that's why. Maybe he can explain on the chats.
Okay, Vatsal, maybe if I don't know how to reach you in this question, because I don't completely understand the question. However, can we do this in the discussion section? Because it's slightly more complex and will take me away from this at this point. Is that okay with you, Vatsal?
Yeah, it's okay with him.
Okay, thanks.
There's
Any
another
other question?
So JD, I have a question.
Yes,
Is Tushar
Tushar.
here?
Yes, Tushar. Yes.
So why in this coupled reaction is it probable that we will see a single hill, that reaction coordinate versus energy plot apart from these two hill shaped curve?
No, it's not necessary. It has to be coupled. The proton and the electron transfers have to be coupled to make it a single coordinate. Okay, and that is what I mean by coupling.
Okay.
That means so who is providing the coupling? The source of the coupling is the molecule itself. Okay, or the reactants itself. Who is providing it? That means somehow the electron transfer and the proton transfer are going through a single degree of freedom. Okay, they're being driven by a single degree of freedom. Okay. It could be an orientation, that's okay, but it's a single degree of freedom, and both electron transfer and proton transfer are happening. Okay, so that's the idea. Okay, so now I'll just move forward because I'm still at a very starting stage. So
so actually we have like already spent 40-45 minutes on the talk, so
oh my god,
fine.
Yes, so this is my trouble all the time. So Sanskruti, could you give me a little bit more time, because I need it to go through, otherwise the discussion will become a little bit difficult.
Okay.
Okay, so so let me just proceed and we'll take the questions at some point. So the thing about reaction coordinates is the definition of it should be given to you, because then it helps you think about it. The reaction coordinate is a pathway along which the reactants can smoothly convert from the reactants to the products smoothly. And that's an important criteria. You cannot have bumps on the way. You cannot have multiple transition states. You have a smooth surface. Okay, and any geometric parameter, any geometric means geometry of the molecule, any geometry or molecules, constituent molecules, any geometric parameter can be a reaction coordinate. It could be just the internuclear separation between the carbon monoxide's carbon and the dioxygen's oxygen. Okay, it could be the angle between two molecules who are present, like this, like my hand, right? The angle which actually sets the reaction. It could be the rotation of the molecules, rotational degree of freedom, or it could be vibrations. Okay, so reaction coordinate descriptors could have different forms. Okay, and I have given you some of these names here. Translational diffusion of reactants, that could be a reaction coordinate. Rotational motion of molecules, that could be a reaction coordinate. Vibrations in a system, that could be a reaction coordinate. And hidden behind these are their intrinsic time scales. That means whatever your coordinate is, the coordinate not only has a geometry associated with it, a sense of geometry, a sense of structure, but it also has a sense of time associated with it, because all of these reactions means motions, right? You have to have motions before atoms can rearrange. So these reaction coordinates have an intrinsic time scales which are hidden in your description. Okay, and this is an example of time scales in a protein. For example, a big protein can have a variety of time scales of motion, and this really dictates its folding and unfolding reaction. The reaction coordinate of a folding and an unfolding of a protein is dictated by the variety of time scales that I marked here. Okay, so you can have, for example, sorry, you can have something like this as a libration or a hydrogen bonding interaction, a vibration, a rotation, a ligand binding, all sorts of time scales. These are the time scales of the constituent motions that are dictating your reaction. So you can see the dispersion of the time scales, 10 to the power minus 15, 10 to the power minus 12, 10 to the power minus 9, minus 6, all in seconds. So this is a femtoseconds, that's picoseconds, that's nanoseconds, that's micro, that's milli, and so on. Okay, so you have so many different motions and a variety of time scales in a protein, for example. So it really is, you can clearly see what is the fastest motion available, what is the fastest degree of freedom available to the molecule? It's the vibration. It's around femtoseconds to picoseconds. That means, in principle, if you were to construct a system and dictate the reaction along one coordinate, the vibrational tailoring of that reaction, of that reactant, would be the best way to achieve the fastest chemical reaction. That means I make the statement, the fastest time scale of a chemical reaction is thus limited by a vibrational period, because that's the fastest motion available to the system. So that means a bond stretching vibration could lead to bond breaking, and that would be the fastest if you could achieve it. Okay? Sir? Yes, Jay.
But vibrational motion.
I don't hear you. Yeah, can you say again?
Vibrational motion is, I mean, as far as I understand, it is very localized in a molecule, right? So for two molecules to come into contact, you will need translational motion, and then that being slower will be the rate determining.
That's right. So if you were to use translational diffusion as a reaction coordinate, absolutely, Jay. Two molecules coming together is one class of reaction. It's called bimolecular reaction. But in many cases, you can keep molecules together. Okay? In many cases, you can pre-organize the orientation of the molecules. Then you can do a reaction faster than the diffusion. The vibrations are much faster, as you correctly pointed out. And this is where I'll do a calculation with you, Jay. See, this is an example of a molecule. You said localized, vibrations can be delocalized as well. I'm going to show you here. This is a molecular system one of my students, senior students, Shunandita, works with. It's called TTF, tetrathiophulvaline. This molecule actually has a very interesting electron transfer reaction, but that's beside the point. I'll come to that a little bit later, but that's beside the point. What you see is, look at this structure of this molecule. It has 4 sulfurs, it has these double bonds, and it's all looking like a conjugated molecule, right? Now, the molecule, as I said, has a beating pattern. The vibrations are the beating pattern of the molecule, right? So you can clearly see what kinds of beating patterns the molecule has. This particular beating pattern is a butterfly motion, Jay, right? It's not very localized, it's the entire molecule moving up and down. This particular motion is just twisting across the double bond of the 2 rings, right? These have their characteristic frequencies. Okay, you can see 27 wave number, it's in vibrational frequency units, 27 wave number, 78 wave number. You can use these frequencies and calculate the time constant of the vibrational period by just doing a simple arithmetic calculation of 1 by c times nu bar. Nu bar is the frequency. Okay, so if you do that, 27 wave number turns out to be 1.2 picoseconds, 10 to the power minus 12th of a second. 78 wave numbers turns out to be 420 femtoseconds. That means these are really very fast motions in a molecule, right? And that's where my entire talk is about. It's these motions that you do not think usually. There is a beating in the molecule that allows for these molecules to, they're always getting stretched out of equilibrium, always getting stretched out of equilibrium. If you can convert those motions into a reactive motion, you can do chemistry. So that's the idea. And you can do chemistry at that time scale. Really, really super fast, right? And there's something else with the vibration that is so beautiful about it. Look at this molecule. It's a bent structure in the neutral, but when it becomes a radical cation, it becomes completely planar. This entire thing is actually bent, slightly bent like this. But when it is oxidized by one electron, a redox, it becomes planar. It becomes aromatic in one ring and it becomes planar. So this planarity actually also influences the kinds of beating patterns, the vibrational patterns you have in the molecule. You clearly see here, there are these two kinds of C double bond C stretches. This one is one, sorry. This one is one. This is one, and that's another one, right? You can clearly see in the radical cation structure, they're very clearly distinct. You can see the end C double bond C stretches very distinct from a motion that is shown here with a different frequency. However, when it is in the neutral form, it is slightly bent. These two motions actually couple to each other and they become mixed. They become mixed modes. Okay, so these are actually going in phase and out of phase with respect to each other. But here it's purely distinct. This is a clear motion on isolated, localized J on this double bond and on this double bond. However, when you have slightly bent structure, they are now delocalized across the backbone. So these are some of the nitty-gritties. There's some interesting things about vibrational modes. They really talk about your system. They are distinct. Every time a molecule changes structure, the vibrational frequencies change. The mode beating patterns change. Okay. So what happens if you could really do this, right? A 400 femtosecond reaction, is it surprising? Well, the godfather of chemistry, G.N. Lewis, pretty much, who did pretty much everything that we know of in terms of bonding theory, he sort of did a very simple reaction. He took this molecule called cis-stilbene and put it under sunlight. And he looked at isomerization of this double bond from cis to trans. And in a 1940 Jack's paper, Journal of American Chemical Society, which is a prestigious journal, even at that point, he predicted in 1940, without any laser sources, he predicted a chemical reaction can have a rate of 10 femtoseconds, 10 to the power minus 14th of a second. And he said it is supposed to undergo through a vibration of the molecule. And this is sort of a very unbelievable number. At that point, nobody knew reaction rates could be in femtoseconds in 1940. And we are 80 years from that point on. But it waited for this prediction to be made true. It waited for something that happened 20 years hence from the 1940 paper, which is Theodore Maiman, an engineer, discovered laser. And of course, with the advent of lasers, everything changed in chemistry, because now you have a light source which is tunable with different colors. You have a light source which is tunable by its brightness. You don't have to rely on sunlight, whose brightness you can never change, right? But you have a super bright source. You can make it really dim or super bright. You can actually tailor these light sources to short pulses. You can do pulsed laser spectroscopy. And you can use these light to also do, say, biomedical biomedical therapies, where you can sort of do photodynamic laser therapy at a tissue where you have a cancer, and you can project your light and have a spatial control on the activity. So in the rest of the time, I'm going to just skim through an example that actually facilitates this idea that vibrational motions, if you keep a tab on, can be the most important way of controlling a chemical reaction, and gives a way to think about chemical reactions. So so this is the heart of this particular picture demonstrates how we actually see by a mammalian eye. Okay, biologists can connect again. Rhodopsin is the most important protein molecule in your eye. that allows for you to tell photons have arrived to the eye, right? How does it happen? How does it do? At the heart of this protein molecule lies the chemistry that dictates photon detection. It's a cis to trans isomerization reaction of a retinal that sort of influences your nerve cells to tell to the brain that you have detected a photon. Okay, and this in itself was a fascinating sort of information that was discovered long back in the early 40s and 50s. But with Jean Lewis's ideas that isomerization could be as fast as 10 femtoseconds, it was worth a mention here that people really looked at this particular reaction for a long time. But how did they start? They had to wait for certain technological advances in the laser science, where people started making pulse lasers. Then they started compressing these pulse lasers to short times, like a picoseconds to femtoseconds. And then you had bright laser sources with femtoseconds. Why do you need these femtosecond pulse compression short pulses? Simply because if your reaction rates are 10 femtoseconds, you need a clock that is as fast as that. So you need something to trigger the reaction watch in 10 femtoseconds. So your laser should be as short as the reaction rates. So this was first demonstrated by somebody called Ahmed Zewail, who got a Nobel Prize. All of these people actually pretty much have been awarded for their fundamental discoveries, but Ahmed Zewail is critical for this talk because Ahmed Zewail did the first experiment to follow reaction rates using light, laser light, pulsed laser sources. And how did he set up this reaction? He basically said, I have a femtosecond pump pulse and a femtosecond probe pulse, which with the pump pulse, I can go up on the molecular excited state. This is now a potential energy surface that you're looking at. You go up the ladder on the higher potential energy surface. You reach this, the molecule starts doing non-equilibrium motion on this potential energy surface, and then it starts dissociating. Okay, and every detail of this was followed by Ahmed Zewail by just looking at just absorption. Okay, the probe pulse would actually look at, once this is excited, what kind of absorption processes ensue. So in fact, he sort of figured this out by looking at time resolved absorption events, that 600 femtoseconds was the time scale for dissociation of a molecule, a simple bimolecular system, ICN, into I plus CN. Okay, similar ideas were later used by Rich Matthews and Chuck Shank. Rich Matthews is my postdoctoral supervisor. I worked with him. And Chuck Shank, who actually sort of used the same experiment that Zewail discovered, or Zewail put together, a pump probe experiment, where now using rhodopsin as a protein system, you excite the retinal in the rhodopsin, and what you do is you go up the excited state, and again you reach this portion of the excited state and you measure the color, the vertical transitions, okay, with the probe. The probe measures these absorption changes. It actually measures absorption spectra of all states that are being populated as it goes into the excited state, and watches the reaction go down to a photoproduct in 200 femtoseconds. This is actually shown here very nicely. You can see the absorption spectrum actually blue shift. Okay, this is the wavelength axis, and you can see the absorption spectrum going blue. Okay, it's going to higher and higher energy. The peak maxima is shifting to higher and higher energy. It shows that the reaction is traveling down to a new product, which is the photo product that you see. Now, this idea was sort of, this 200 femtosecond reaction was then followed up and went to shorter and shorter pulses, which sort of talked about the beating patterns. It enumerated the beating patterns of the molecule. And these beating patterns resulted in the discovery that this particular reaction is driven by few reactive modes, and those reactive modes were sort of later on enumerated by a group in Germany in 2015 by the name of Duane Miller, who said that there is a double bonding torsion, the double bond torsion. So the torsion is something like this. You have this double bond and you twist the molecule. That's a torsional mode of a molecule. It's a vibration. It's a vibration in the system that twists the molecule across this 11-12 double bond, through which this isomerization can happen. And it turns out the product came out around 50 to 200 femtoseconds, and the vibrational mode time scale, which is a 568 wave number mode, the torsion, appears to have a vibrational period of 58 femtoseconds. Tells you that within 2 or 3 vibrational periods, the reaction was done. And that in itself was a major milestone in our understanding that chemical reactions, if engineered in such a way that the reaction coordinates, okay, can be driven in such a way that you have a very fast reaction almost at the vibrational time period of the reaction, then that reaction will be highly efficient. The photon energy is completely dumped to this vibrational mode, and that gets activated 4 times before the reaction actually happens. It's actually this motion. It goes 4 times to a torsion, and then the product forms. So this is absolutely stunning result, simply because it tells you that you can force your system. You can force your system to do this. A protein molecule does this, and that is the essence of how nature works, right? A protein can actually pre-organize the reactant in its active site to make that reaction happen so fast, so with so much rapidity. There are more information that I'm not going to discuss now because we have run short of time, but there was very interesting observations in the 2018 paper, which showed that not only vibrations like torsion that are important, but there are other kinds of motion which are participant to this reaction, which are called the hydrogen out of plane motions. So any double bonded system will have these hydrogen atoms, and especially this molecule has these hydrogen atoms, and they can move out of plane, okay, like what has been shown here. And these oscillations of out of plane, they can have in phase and out of phase oscillation. These 2 vibrations could be in phase, these 2 vibrations could be out of phase. That means I'm twisting in one direction, then the oscillation of the hydrogen out of plane should be in the same phase as the twist. If it is in the same phase, you get a successful reaction very fast, 200 femtoseconds. If it is not in the same phase, you never get the reaction. So this really tells you that the beatings, the heartbeat of the molecule, really, there are so many different kinds of heartbeats, so many types of vibrations associated with the molecule. They have to be in phase for the entire reaction to take place. This is a stunning result, absolutely stunning, because you can imagine, it actually tells you, if you know coordinates that are driving a reaction, and you can engineer a reaction to be very fast. This is that was an example of isomerization reaction. The GFP molecule, which I think the biologists here have very good idea. The GFP molecule actually goes through an acid-base reaction in its excited state. And that's why the green color comes, because the deprotonated form fluoresces. The protonated forms is the neutral form. It goes up in the excited state. It deprotonates in the excited state, and then it emits. So the GFP has an excited state acid-base chemistry. And believe me or not, and that happens, okay, in tens of picoseconds. It's much slower than what the vision protein does, but experiments have identified a reactive mode that is 280 femtoseconds in lifetime. So it requires 30 vibrational periods to reach the reaction, to finish the reaction of GFP. That clearly tells you that it has to wait. After this reactive motion, which is motion of a ring that is moving up and down, the reactive end is somewhere here. It's an OH bond that is breaking the proton transfer to the water here. That's the acid-base reaction we are talking about. This particular ring actually keeps moving up back and forth, till it finds a right orientation with respect to the water molecule, the proton gets dumped off. That's the reaction that the GFP does in tens of picoseconds, although this reactive motion is 285 per second. It really has to wait for the water molecule to really orient itself. Okay, and I'm going to finish by saying that we in the group have been looking at a combination of proton transfer and electron transfer reactions. And in fact, we've been successful in doing very fast H-atom abstraction reactions in few hundreds of femtoseconds. Again, vibrationally dictated. We have not been able to identify the reaction coordinate yet. That's a work in progress, because these are very, very fascinating experiments and difficult experiments to do in the lab. But that's something that we are doing. We are looking at vibrational snapshots of these systems in the lab. And since all of you at CEBS are in the same city, okay, you are more than welcome, if you have interest, to look at the technology that we have to look at these vibrational snapshots in the excited state. We actually directly measure these vibrational beating patterns as reaction happens. So, so these are some of the things that we typically do. I could not enumerate out our particular method of doing H-atom abstraction, but maybe later. But at the end, I would like to give a summary of what this perspective is all about. So when we look at reactions not from the classification of electron transfer, proton transfer, or redox, acid base, or rearrangement, we can go beyond that classification. And when we look at the reaction coordinates, we are actually addressing every molecule separately, every reaction separately. And it's important to understand that the vibrational beatings or the ticks in your molecule, if you keep a tab on them, if you can engineer certain reactions which actually use those motions, you can create systems which are very, very highly reactive, very fast in time scale, and highly efficient. Okay, so this is where I would like to state that the chemical reaction clock, which I meant, was nothing but the motion, the time scales of the vibration in your molecule. The unit of it is dictated by how many vibrational periods is required to do a chemical reaction. So that clock is the one that you would like to elucidate for every system. If you could design newer classes of reactions, and the implications are immense. We can have new kinds of solar cells. We can have new kinds of photocatalytic approaches, and all of them happening with very high efficiency, energy efficiency. Some of this idea, which I have left out in the open for a discussion, which is called the coherence or the coherent chemistry, is actually utilized by photosynthesis. In some sense, people have looked at that as well. So I would like to stop here and say that there is a new way to look at chemistry and a new way to plan reactions, and it is to go through the reaction coordinate picture. If you can identify the modes that you can use, their time scales, you can drive chemistry at time scales of your choice. And by rule, if you can do your reaction within 1 to 5 vibrational ticks, you can define that chemistry as a coherent chemistry. If you go beyond 10 ticks or 10 such beating patterns, you might lose energy and you might go in other surfaces. So it's very interesting to look at these coordinates. With that, I would like to thank all my group members for being part of this endeavor, and all these members. And you can see your own friend Gursayeb right here, and I'm so happy holding
Kiran also.
that's one of those group pictures we took. And you're free to visit our website or email me for questions in future regarding the talk. And thank you all. Sanskruti, thank you. I'm sorry for taking, as I had actually warned you, I might take a little bit longer, but that's what it is. Yeah. Thanks, Sanskruti.
So we can take questions.
Yeah.
So can I ask?
Yes, Jay.
So for a lot of these things you mentioned, it takes 1 to 4 vibrations for
Yes.
the reaction to take
Yes.
place.
Yes.
So yeah. So I mean, why is that number fixed, and why does it take multiple vibrations for a reaction to occur?
That's right. That's right. So that number is not fixed. What I would like to say is your reaction could be driven by any of the vibrational modes in your system. Now, every vibrational mode has its own characteristic time scale, right? So if I were to give a number that this is a definition of coherent chemistry, that number has to be universal across, independent of the time scale of a vibration. So hence, when I say coherent chemistry means, 1 to 4 vibrational periods or 1 to 5 vibrational periods, it just normalizes all vibrations to 1 unit of a vibrational period, and it removes the numbers for the reaction. Okay, so there is no fixed number. You can drive your reaction by a 280 femtosecond mode. You can drive a reaction by a 100 femtosecond mode. You can drive a reaction by a 10 femtosecond mode. It depends on how you have positioned your system, and slightly different way of thinking. Typically, when I say my reaction is efficient because it goes by this time scale, it doesn't matter. What matters is who drives the reaction, and what are the fundamental time periods of that reaction coordinate. So if it is a translational diffusion, it will be nanoseconds, Jay, right? As you mentioned once, right? It will be nanoseconds. So if it is translational diffusion, I'm never going to achieve femtosecond chemistry. Okay. If it is vibration, you could. If it is rotation, you can do picoseconds. So those are some.
So my question was more like so you have, say, a bending vibration, right? So every time it bends, is it the same, or is the bending also different with each?
Yes. So every time it bends, if it is not reacted in that, it has to come back in the harmonic way back to its original position. It is still the same molecule.
No, like suppose I have the molecule, it bends, for example.
Got it.
So the amount it bends isn't the same, because if that is changing, then this might cause this discrepancy. Otherwise, it
right, Jay. Yeah. So we calculate vibrational frequencies for minimas of stable geometries. Okay, when the molecule is undergoing a change, okay, as the molecule is structurally undergoing a change, there are no well-defined vibrational frequencies for those. So what you do is you actually have a memory. You have a memory of the stable structure. So you are driven by that vibration. So this particular, say any surfaces that I draw here, I'm going to erase these. So yeah, I'm going to erase these. Okay, so here you see a harmonic-like potential I've drawn, Jay. This is a minima. This position, the geometry of the molecule in this minima has a well-defined normal mode pattern. Okay, this is a minima in the excited state. Okay, so again there will be a well-defined normal mode, but these are the motion. These are the places where the structure is in non-equilibrium, right? So the only residual is coming from the memory of the original vibrations that was pumped. With the energy was the photon were given, and you drove these molecules to an excited state. Okay, that excited state has its own vibrations defined, but this molecule has its own geometry which was pumped up, right? So it goes with its own vibrations initially, but then it readjusts on this potential as it is changing its geometry. Do you understand? So those vibrations will come to fore only when it's achieved its equilibrium geometry here. So suddenly the vibrational frequencies will change, okay, slightly. So does that answer you? But you have a memory. You have a memory of the original state's vibrations, and that drives you to the reaction. And that is why this is the coordinate which you start defining. That's a good question. Yeah.
Vatsal also has a question. So he's asking that, so there are photons of many energies in the visible spectra, but rhodopsin is molecule which has only one kind. So how come all the wavelengths in the spectra manage to trigger the isomerization reaction that you were talking about earlier?
Vatsal, actually that's not true. I have not shown the absorption spectrum of rhodopsin, but I can draw it for you. The absorption spectrum of rhodopsin goes as this. It actually is centered at 500 nanometers. My drawing is really bad with this pen, but this is 500 nanometers. And I'll tell you what the span is for this absorption spectrum. It's close to 50 to 100 nanometers, okay? Like 100 nanometers. So that span means it goes all the way from 350 to 600, okay? So anything under that absorption envelope will be activating the isomerization reaction, because that's the span of absorption for the retinal molecule, okay? However, your eye actually sees all sorts of colors. So it's just not rhodopsin. Rhodopsin actually is not part of color vision. Rhodopsin is part of the dark vision or the unicolor vision that you have, okay? It's only to say this is a bright room and this is a dark room. There are other kinds of opsins that absorb in a variety of regions of the absorption spectra. There are green opsins, yellow opsins, red opsins, okay? So they are distributed, and your eye has a different detector for these kinds of photons. Rhodopsin is only for distinguishing dark versus bright room. Okay.
Okay, JD, I have a question.
Yes.
So basically, the time scale for that torsional motion, is it dependent on the length of that conjugated chain, or else it is independent of that?
The time scale of the
Torsional motion, that torsional motion.
Torsional motion, that's a great question. Yes. An ethylene moiety, Tushar, if I go here and draw it,
yes,
ethylene moiety has, ethylene is HH, all H, correct?
Yes.
All H. So an ethene, as we define, this has a torsional frequency depending on the corresponding masses of atoms associated with it, right? Because it depends on the reduced mass,
yes,
correct? So in this case, and of course the force constant. So in this case, this would be close to maybe at the top of my head, I would say like 600 to 700 wave numbers. But when it is part of this entire chain, both the reduced force constant, the force constant is the bonding strength, as well as the masses associated with the carbons, right? They change. And so the torsion will become slightly different in frequency. So it does have a context. It depends on where is the ethylene group positioned, in which kind of system. The torsional motion will be different. The frequency will be different. And the normal mode could be different. Okay.
And one thing, the time scale for that torsional motion from changing from cis to trans, will it be equal to the change from trans to cis? Or
no, that's great. Again, no, it's not going to be. That torsional motion has a different frequency. As well as it does not go through the torsion. Actually, the cis to trans goes through both torsion and the hoop. But sometimes in other cases, it may not be just the torsion. There could be other reactive modes as well.
Okay, okay, got it.
Sir, this is Prithvish.
Yeah, Prithvish.
Yeah, actually, I was hoping that you could explain again what is the significance of the length of the pulse.
Great.
Yeah.
Great question.
Yeah.
Yeah, I had to go through very fast. So Prithvish, if I have to observe, okay, a reaction which is 10 minutes in origin, what will you do in the lab? So you will take a stopwatch,
Yeah.
correct? What is the time unit of the stopwatch you will take for a 10-minute reaction? Will you get away with a 1 hour time resolution for that stopwatch? No. Right? You will get away by maybe seconds. If you have a second stopwatch, you might be having a good time resolution to follow that reaction in few minutes, right?
Yeah.
What if your stopwatch has a millisecond time resolution? It's an overkill. It's an overkill, right? That's exactly the idea. When G.N. Lewis predicted 10 femtoseconds for an isomerization reaction, all these folks down here, mentioned here, really tried to go around and make the stopwatch, that is the laser pulses, go to very, very short time, at least to 10 femtoseconds. If I have to watch a reaction which is 10 femtoseconds, I should have a pulse that has a width of 10 femtoseconds or shorter, right? In fact, we were lucky that it's the Rhodopsin reaction goes 200 femtoseconds. G.N. Lewis was slightly off in this isomerization time scale, but it's around 200 femtoseconds. Okay, so
it's about observation of this.
It's about observation. So you have a short enough pulse, excite the system, and watch the system. You have to use both two pulses. Excite faster than the reaction, watch faster than the reaction. It's a pump probe technique.
Okay. Thank you.
I think Chandra had a question.
Yeah. So when you mention 1 to 4 ticks or 5 ticks, 10 ticks, so we are assuming a phase locking kind of a thing, right?
Great.
So how do you just assume that, or is there a way you can?
So the phase locking means the coherence, actually. Okay, coherence, the word comes from phase locking of the motions. When I say coherent chemistry, it means there are certain vibrational modes that are coupled in phase to drive chemistry. Not one single vibrational mode can actually, typically not just one single vibrational mode drives chemistry. Okay, there are a couple of modes. And if they are locked in, this is an example of coherence. See, this is a beautiful illustration of vibration phase. Okay, they have to be locked in to do reaction. This is a stunning paper, by the way. If you guys have time to read this, this is a simple experiment they did with the deuterium isotope effect. They replaced this H. So what they did was, again, looking for the pen, they replaced one of these with D. They replaced both of these with another experiment. They replaced both of these with D. And then they measured with respect to the original proton, diproton substitution, right? And they found out that when it is a single D, this is out of phase. The reaction rates are different. Okay, it's a beautiful experiment of phase. Okay, just a slightly heavy atom, and you disturb the phase. So this was done by Phil Kupura and Mathis. But you know, that's the idea that these motions should be phase locked to do reactions.
So and then in the last slide you had mentioned that after 10 ticks or so, it doesn't is not coherent anymore.
Yes.
So,
this is a bold statement. I made this statement simply because you need to have some idea what it means, what coherence means. So just let's assume this. The most local bond stretches are the CH stretches in an organic molecule. Okay, CH. Now these CH vibrations have a vibrational frequency of 3000 wave numbers. If I do the math correctly, convert 3000 wave numbers to time period, you can get this, like it's a 13 femtosecond time period. Okay, so the most highest frequency vibrational normal mode in an organic molecule, which is a CH stretch, it actually has a time period of 13 femtoseconds. Okay, now these 13 femtoseconds, you can do the fastest reaction you could do is a 13 femtosecond bond breaking, one single. Has anybody done it? No. We have done close to 14 picoseconds. It's 3 orders of magnitude slower than that. Okay. But when I think of that question, that is where this makes a difference. If I know that the fastest mode actually is 3000 wave numbers, 10 femtoseconds, and I try to do the reaction at that limit of 10 femtoseconds, I know that a single vibration led to that, right? A single vibration period led to that.
Yes.
And it can only happen if energy was really dumped into the system right at that spot and the bond broke, correct? That's how you can get it. This direct coupling to that mode. But the problem is when you dump energy to any molecule, it does not go to one mode. It goes to multiple modes. This is called intramolecular vibrational relaxation or energy redistribution. Okay. What that does is when I excite the system, I give energy to the entire molecule, and the vibrational energy gets redistributed in many modes. Okay. Statistically, that's what you do. You look at statistical mechanics. It's a very nice statistical mechanics problem where you dump energy of equal to E and you partition the energy, and partition it equally. It's called equipartition theorem. Okay.
Yeah.
You partition to all sorts of modes in the system. However, we don't want equipartition. We want energy to be dumped in specific modes. And sometimes you can do that, achieve that by going to systems like this, where molecules are pre-organized with respect to each other and already ready to react. Okay, that's what we do. This is actually a diagram taken from my student Dipin Tomar's rendition of how we would like to do photocatalysis with light at very fast time scale. So this idea that you need to be few reactive vibrational ticks, that is coherent in phase locked chemistry. Now, if you go beyond 10, the reason I'm telling you that it's going to impede is simply because by the time you are doing this, you are redistributing energy to all modes, and hence thermalization will take place. Okay.
Fine. Yeah.
Sir, generally, Abhi, just you talked about these modes, the energy getting redistributed in multiple modes. So I wanted to ask that whatever I understand about statistical mechanics is it is defined for a category of particles, right?
Right.
Yeah. So is it similar to phonons? I mean,
yes, it is. It is in some phonons are well defined in a solid state system.
Yes.
So in molecular system, these are normal modes of vibration, right?
Okay.
So yes, it's very similar. You basically give energy, you hit it with thermal energy or a photon energy being kicked, being given, and you redistribute the energy. Not only you go to the excited state with the photon, but you redistribute the excess energy amongst the vibrational parts. Okay.
So hello sir, I have a question.
Yeah.
So I am asking about that when you give the light to that rhodopsin molecule, so specifically why the 11-12 double bond is why isomerized, not others?
That's a great question.
The molecule take the whole energy of the photon.
Yes, Pradeep. That's a great. This is exactly what the protein does. It's called pre-organization. Okay, what it does. Look at this molecule. Again, I have to. Okay, I'll go to the front end. Yes, here. Okay, look at this molecule, Pradeep. So there is a methyl here, Pradeep. Can you see the methyl here? Pradeep, are you here? Pradeep, are you listening? Okay.
Yes.
Yeah. So look at the methyl position, Pradeep. It is next to a hydrogen here.
Yes.
This is a hydrogen. So what that does is there is a steric clash, and the protein allows in this entire retinol to be positioned in the active site in such a way that it allows for a slight bend in this double bond. That means a slight distortion in the double bond because of that steric clash. And once it is slightly distorted out of plane, it is ready to zoom into the torsional mode, because what happens is in the torsional potential, this picture looks like this. It goes up vertically, and it goes down with a strong force. There's this large slope. There's a lot of large non-equilibrium motion that has happened for this state because the atoms are completely in a different geometry. And then they rush back. They rush back because there is a harmonic potential, a new harmonic potential they have reached. They try to rush back, and that forces the reaction to happen. This is a torsional potential, and the torsion allows for the bond rotation. Do you understand, Pratik? It's only this particular mode that is displaced, and the protein allows for it.
Okay, so the methyl hydrogens, the clash you saying, so this is also occurred in the femtosecond range.
So this, yes, this torsion is 58 femtoseconds.
Okay, or I want to ask that when the molecule goes that state and the double bond, that is the isomerization is occurring, so the other double bond has any effect on that time scale, that is their tensor or their polarizability?
That's a great question, but what has been measured is that the double bond rotates and the other double bonds are pretty much the same. Okay, there is of course computations done. Here this is shown. Pradeep, could you see?
Yes.
Yeah. So you see there is a torsion here, and there could be torsional motion as well here. Okay, Pradeep. However, this dominates, and it just takes out the bond and it reorganizes. All the atoms have to reorganize, Pradeep. Of course, they have to come to an equilibrium. The rest of the molecule. So they will also adjust, but they are spectator modes. They are not the reactive mode.
Yes. Okay. I understand.
Hello, sir. This is Subhajit.
Yes,
sir. I have a question. How do you ensure that the probe laser doesn't affect the reaction itself?
Fantastic question.
And also, what should be the pulse frequency of the probe laser so that you can measure it with higher accuracy?
Thank you, Shubhajit. Thank you for asking those questions. The probe laser is at least 100 times weaker than the pump laser. Can we? Is that okay with you, Shubhajit? So
yes, sir.
Yeah. So we don't use the same kind of intensities for the probe laser. And when we pump, so if rhodopsin is something like this, Shubhajit, we pump it with a laser which has a color that can pump the main absorption feature. This is the absorption feature. That's your laser spectrum. Okay, that's your pump.
Yes.
Now the probe is, however, I will change color. Can I change color of the pen? Maybe not. Okay. The color of the pen. Okay.
The ink color.
The probe is something like this, Shubhajit. It's probing everything. It's a large, broad bandwidth, very weak pulse. Okay. And it probes so that I can measure absorption spectrum. Like your lamp in the absorption UV spectrometer, it probes a wide range of wavelengths. Okay.
Okay, sir. And sir, what should be the frequency of this pulse so that it doesn't interfere, like you can measure it?
Frequency of this probe pulse?
Yes, sir.
So the frequency is distributed, as you can see. There are multiple wavelengths that are there in this probe.
No, sir, the pulse frequency.
Huh?
The pulse frequency.
Oh, the pulse frequency of the repetition rate of the pulse. Okay.
Yes, sir.
So you can choose any repetition rate as long as your sample is not getting damaged. So typically when we do in our lab, it's a kilohertz. But you can go down to few 50 megahertz or a megahertz rep rate. Okay. It just depends on the technology of the laser that you're using. However, it should ensure that you would give enough time for the molecule to recover. Okay.
Okay, sir. Thank you, sir.
So I think we should go towards interactive session now.
Okay.
So
thank you once again for this opportunity. Thank you, guys.
Thank you, sir, for coming.
Okay.
So
I had one question.
Yes.
Okay.
So if I get you right, there are these fast vibrational degrees of freedom in your molecules.
Yes.
And like for every molecule, these vibrational degrees of freedoms are different because each molecule has a different structure.
Own set.
and you are trying to excite these degrees of freedom. If
That's
they
right.
get excited, the
Yes.
reaction goes ahead. If they don't get excited, then
Okay, can I just take a brief break from that question, and I'll just say this: it's not just the excitation that matters. It's not just the excitation that matters. It also matters what is the context of the molecule. The molecule could be positioned inside a protein pocket. The molecule could be free in solution. The molecule could be in some other context, chemical context, right? That molecule will have the same set of normal modes. Okay. However, with the context, these normal modes could be engineered slightly. Okay. And that's what we are trying to do. You would, for a chemical reaction, not only you need to excite normal modes of the molecule, isolated molecule, but you need to place the molecule in a context. And that's really important. That's the idea what enzyme does. It pre-organizes. This retinal was in a context of the protein pocket. So it's not just that अरे excite कर दिया यार वो सारे modes को and reaction हो जाएगा. नहीं, reaction नहीं होगा because mode there will be equipartition theory also. So you have to beat the thermalization and put the molecules in a right, the other molecular partner in the right orientation, so that you can harness those excitations in the right time scale.
okay, so what does the environment, like if you change the environment, what does that change? It changes the coefficients, the different vibrational
So
coefficients?
it changes the frequency, okay, of the motion, which hence changes the time period, right?
Okay.
And it also changes it adds a new set of coordinates which come with intermolecular interaction, a molecule interacting with another molecule, right? So if there are two molecules and you want to do a reaction between them, right? So the molecules have to interact. You have to pre-position for any kind of reaction that is coherent. I don't care about if it's not coherent. If translational diffusions bring the molecules together, all of these bets are off. We are not doing fast chemistry, okay? But if they are together in one place, in one way, in one orientation, they have an interaction. If you set up an interaction, there's a normal mode. It's an intermolecular normal mode.
So are you saying that the nature of the degree of, the nature of the reaction coordinate itself changes?
Yes, it does.
That was your latter point, I guess.
Exactly, it does. But it will have bearing on the original vibrations. Absolutely. The composition of the vibrations will, you can still excite the same kind of mode, okay, that you were doing in free solution or in isolated. But now you've slightly engineered and you push the reaction. That's the idea. You do not let thermalization take place in an isolated molecule, which is always taking place in an isolated molecule.
Okay, so you change the environment to ensure that thermalization does not take place. Okay.
Exactly.
Okay.
And that falls into chemical intuition, right?
Okay.
For eons, chemists have been using chemical intuition to predict reactions. How do they predict? Bring molecules closer, have a orientation, right? Have a functionality. Like a OH group has a very specific reactivity as compared to COOH, that means an acid functionality. How does one conceive all those things? Because they know that OH arranges well with another, say for an O minus or something, right? So they will have a well-defined chemistry that intuitively I can predict. So this is actually falls back. You can project all these back to the reaction coordinate picture and normal modes. That's the idea that I'm trying to stress upon to everyone. That our intuitive field of chemistry has deep-rooted, deep-rooted from the fact that we have learned empirically, but they all actually can be explained in this picture.
Okay.
Yeah.
But I'm trying to get a idea. Some understanding of what you said about 1 to 5 vibrations.
Periods.
I mean, yeah. If so what was that again? If the
so
bond
the idea
breaks,
so the idea is, my proposition is that if you were to do a chemical reaction within 1 to 5 vibrational periods of your reactive coordinate, okay, of your reactive coordinate,
somehow,
but it doesn't matter, then you are pretty close to the highest possible energy efficiency, because you have done something called as coherent chemistry. You have actually ensured that the vibrational energies that you have put in the vibration modes, right? That is letting out, that leads to a reaction. And now reactions will have their own energies, right? Products will have their energies, right? So you are actually creating creating situations where the fragments coming out of that reaction, right, will be as excited as the original reactants. You are not going to thermalize as soon as possible.
Thermalization happens.
Then you can thermalize later. Okay. You can dump that energy into other modes. Okay. But initially, all of this has been focused on one single mode. That's how that 1 to 5, you have enough time to do this, right? If it is 1, for rhodopsin, it's almost 1 to 2. It's just set up to do the reaction. There is absolutely no way you only lose energy in only one part. That is the molecule has to come down the potential. Okay, that is where you lose energy. But it's all done within few 200s of femtosecond.
But if say there are like 10 vibrations, that means you're losing a lot of energy in
absolutely. So then you are delayed. I don't know how long. You've exchanged energy between modes and you've done lots of other things.
Okay.
Because when it comes back, remember, when some harmonic motion is coming back, it is still coupled to the original set of oscillators, right?
Yeah.
So you can still do energy exchange during that.
Yeah.
But if you do it with one go, no energy exchange happened. You just did, simultaneous, right?
Yeah, the other degrees of freedom are much slower.
Yes, exactly. That's the idea.